This video is the last installment for our first topic for exam for ATCase. And what I want to do here is just go over some of the figures that are in the textbook. This is figure 10.11, 10.11. And what's illustrated here is a comparison of a regular Michaelis-Menten enzyme, shown here in blue, with an allosteric enzyme like ATCase, shown in red. And what the comparison shows is the degree of change in concentration for the substrate that's needed to go from 0.1 maximal velocity, here on the y-axis is maximal velocity, fraction of maximal velocity, to 0.8. So how quickly can we go from relatively low rate of processing our substrate to a high rate by changing the concentration of substrate? For a Michaelis-Menten enzyme, you have to increase the concentration of substrate over a relatively broad range of concentration. 27 is highlighted in this particular figure. For an allosteric enzyme, the degree of substrate concentration change is much less, and we call this, we describe the situation as the allosteric enzyme has a threshold, is much more sensitive to the subtle changes in the substrate concentration compared with a less intelligent Michaelis-Menten-like enzyme. So this is an example of one of the clear biological advantages of having an allosteric enzyme in place instead of a regular, less intelligent enzyme. Another place where biological advantage becomes clear with the allosteric enzyme, we're not just sensitive to concentrations of substrate. Here, this is figure 10.14 from the textbook. We're also able to adjust to adapt to different conditions, different cellular conditions. In the case of a cell that has a lot of resources, that's going to be reflected in high levels of ATP. And now we can make the enzyme spend the ATP more aggressively by shifting this curve, this rate of the reaction as a function of aspartate to lower concentrations of aspartate. If we have if we don't have ATP and the energy reserves are lower, and also if we have sufficient product accumulated in our biosynthetic pathway, now we can shift this response curve to the right, meaning we need much more aspartate in order to achieve maximal velocity. So another biological advantage for incorporating allostery into the system is that we can make the enzyme sensitive to more than just one input. It's not just responding to concentration of aspartate, but it's responding to these allosteric effectors, ATP and CTP.